sir. Yes.
Please continue. Madam, please continue.
Yes, sir. So our next topic is actinomycetes. These actinomycetes, you can understand like these are filamentous bacteria and they stain gram positive when you apply gram staining technique. Okay. So important point here is they are filamentous bacteria. Next. They are having characters common to bacteria and fungi. Okay. We read about linking organism. Similarly, these actinomycetes, they share features with bacteria and fungi. And earlier actinomycetes are clubbed with bacteria in the same class, that is schizomycetes, but they are confined to order actinomycetes. So classification wise, they were kept along with bacteria. And this mycelium is unicellular and it is non-septate, means cell is single cell and cross walls are not there, it is a non-septate and it is a cenocytic, means multinucleate structure. Okay, and the growth is very slow. They show distinct colonies which are powdery in nature. Okay, these are about the role of actinomycetes. Actually, we are getting many antibiotics from this class of organisms. Okay, whether it is Streptomyces or other organisms, they belong to actinomycetes only. And some of the main functions are they degrade all sorts of organic substances. So they are helping in degradation and whatever organic residues are there, first they will be attacked by bacteria, then fungi, and then by actinomycetes. They produce dark or brown pigments, black or brown pigments that gives color to humus. You know about humus? After degradation, humus will be formed in the soil. So these organisms, they contribute to the formation of humus and they give that dark color to the humus. So these are actinomycetes. The colonies will be dry and powdery in nature. Coming to the characteristics, as we discussed, they are mycelia or filamentous in nature. Examples are Streptomyces and Nocardia. They form spores. Spores are resistant, the structures which can overcome unfavorable conditions. And one of the representative of this group is Streptomyces. And under Streptomyces, we have around 500 species. And these filaments, in these filaments, branching is seen. Growth is always at the tips of filaments. From the edge of the filaments, growth will happen. And vegetative phase is having a complex tightly woven matrix. As we have discussed, these are filamentous cells. Okay, so they are woven with each other, interwoven, and they form a compact mycelium. As a result, colony it looks like a colony. As the colony ages, aerial filaments called sporophores are formed. So up on the at the top, sporophores will be formed. The project above the colony and give rise to spores distinct from endospores. So some spores will be formed and we can see them. Okay, so coming to the growth of this, first from the spores, they'll germinate and they'll form this filament, and then this mycelium will be formed, and then aerial growth. One second. First, the growth will be at the base here. First, the growth will be at the base, and then aerial growth will happen. Last is sporulation. This is actinomycetes on plate. The colony will be dry in nature compared to normal bacteria. These are all different types of spores. See, this is the formation. Normal bacteria will be just present on the surface of the agar. Okay, here we are growing actinomycetes in lab, in the media, right? So the roots, these structures, whatever present in the subsurface area, they will give more strength to the colony. So we can't remove that colony very easily from the plate. It will be very tightly stick to the colony, that media. And ecology, they are present in aquatic habitats, and they are more present in soil, and they produce geosmin. You know what is geosmin? It is rainy season, right? We get good
Yes.
smell of
Yes.
soil.
Yes, madam.
So that is due to geosmin, and this geosmin is produced by Streptomyces species. They prefer alkaline to neutral soils, and nutritional requirements are different for different organisms, and they produce extracellular enzymes. As we have discussed, they carry out degradation of organic material. So for degradation, different enzymes are required, and 50 different carbon sources can be utilized by these organisms. And as I said, Streptomyces, they are helpful in antibiotic production. The antibiotic produced by Streptomyces is streptomycin, and over 500 distinct antibiotics have been produced by this single genus. 500, the number is very high, right? More than 60 antibiotics have been produced which have application in human and veterinary medicine, agriculture, and industry. So now, like, we are getting many problems day by day, right? We get cold, we get fever, whatever be the problem. If we get wound also, to come out of it, we need antibiotics. Yes or no? Without antibiotics, treatment will be very, very difficult. And nocardia. So first organism we studied under actinomycetes is Streptomyces, and the next one is nocardia. This is also a mycelial form, and they also form aerial spores. Sometimes they are acid fast. Sometimes they are acid fast means lipid content in the cell wall is very high. You know about Mycobacterium tuberculosis or Mycobacterium leprae. Have you ever heard? They will also have acid fast cell wall. Means their cell wall will have more lipid content, and they won't allow the stain to enter into the cell. When we are talking about gram positive or gram negative, we are adding some stain to the cell, to the bacteria. Okay, first the cell should allow that stain to enter into the cell wall, otherwise the cell won't get stained. So similarly, nocardia, some cells they will show acid fast nature where more lipid is present in the cell wall, and these cells will not allow that stain. And common soil, these are the common soil organisms, they are found everywhere, and these are obligate aerobes. Obligate means compulsion, obligation means compulsion, obligate aerobe means compulsory aerobic in nature, means if you won't give oxygen they will die. They need compulsory oxygen, and many of them utilize hydrocarbons, and they weakly stain gram positive. Why they are saying weakly stain? Because due to presence of more lipid content, it cannot take more stain. That is why it is weakly stained or showing very less purple color. That is why it is called as weakly staining. And catalase positive rod shaped bacteria. Catalase means hydrogen peroxide breaking capacity. It forms partially acid fast beaded branching filaments, but being truly bacteria. If we see the structure, we feel as if it is a fungi, but it is a bacteria. It is having total 85 species. They are non-pathogenic, and some are responsible for nocardiasis.
Ma'am, catalase positive once again.
Catalase positive they react with hydrogen peroxide. If we add that culture to hydrogen peroxide, oxygen will be released. Reaction, it is a biochemical test. You know, different biochemical tests are there.
Yes,
In
ma'am.
the previous slides, we have discussed about glucose utilization. Similarly, catalase test is also a biochemical test where you will add this culture to, culture means the bacteria, whatever you have grown, that culture to hydrogen peroxide, then you will get effervescence. In chemistry, you will come to know bubbles.
Yes,
So
ma'am.
that bubbles will be coming. That bubbles are produced due to release of oxygen because hydrogen peroxide will be broken down. Okay.
Okay.
Next, nocardia are found worldwide in soil rich in organic matter, and they are oral microflora found in healthy gingiva as well as periodontal pockets. Means as we have seen normal flora. Okay, so this nocardia is a part of our oral cavity. They are present in mouth, in different parts of our mouth, and these infections are acquired by inhalation of the bacteria through or through traumatic introduction. If we inhale nocardia, they can cause problem, or through traumatic by accident they can enter into our body. These are all actinomycetes, and then so nocardia also they are causing pneumonia. Some are causing endocarditis related to heart, and they can also cause encephalitis or brain abscess. Even they are causing cutaneous infections, that is skin. These are some of the diseases caused by nocardia. One is pneumonia, second one is endocarditis, third one is encephalitis or brain abscess, and then cutaneous infections. Okay, see this? This is due to nocardia. So here we end this lecture of actinomycetes. So with this we have completed the list of topics given by Tirupati sir. Sir,
Yes, madam.
I think we are done. Along with this, one more topic sir suggested me to take, that is economic importance of bacteria.
Yes.
Just
Yes.
there, nothing will be there to explain. In Google, if you search economic importance of bacteria or microorganisms, you can get the list. Okay, try to remember whatever possible, because everything you can't remember. Whatever is possible or whatever is famous, you feel that you can remember. That's all, sir. We can stop the recording.
Okay, madam. Sarya ji and Naresh ji and Renika ji, Amrin ji and Prakash ji, Bhavani garu, any doubts?
Once show the actinomycetes, that spore slides, sir. Spores.
Uh-huh.
Before this, ma'am. Before this. Before this, ma'am. When the spores change into germination, that arrow.
This one, ma?
No, ma'am. Before this. Upside only, ma'am. Upside. Still more upside, ma'am. Yes, ma'am.
This one?
Ma'am.
Huh. You have a doubt here?
Once you say, ma'am.
You want me to explain this slide once again?
Yes, ma'am.
Okay. So here it's a cycle of growth of this Streptomyces. First, we start with spores. Okay, these are the spores. Spores will germinate. Okay, after germination, they will form the vegetative mycelium. Mycelium will be formed. Okay. After that, aerial hyphae, as I have shown in one slide, first growth will be under the surface, subsurface, then on the above the surface. Okay, so first vegetative mycelium will be formed downwards, and then aerial hyphae will be formed, and then from this hyphae spores will be developed. Okay, again these spores will germinate and they form mycelium.
Yes, ma'am. Thank you, ma'am.
Okay.
Right. Madam Ji, really wonderful session. All of our participants as enlightened from your class, madam. Very
Actually,
briefly, madam.
we could not get into more depth of these topics because these were not frequently asked. The questions were not frequently asked from this area. So అందుకే అంత డెప్త్ లో చెప్పలేదు సార్, పైన పైన చెప్తే సరిపోతుంది.
Okay.
వేరే బాటనీ టాపిక్స్ చాలా ఇంపార్టెంట్.
Okay. If any possibility, take one or two classes for the in the practice, like in previous classes, madam. Our students is asking me.
Sure, sir, I'll try.
Okay, we'll inform our students. Okay,
Yes.
madam. Thank you, madam. Thank you.
Okay.
Right, madam.
Sir, thank you.
Okay. Prakash.